results for those settlement experiments after exposing larvae to those dishes of bacteria, dishes of those monospecific biofilms. On the x-axis are the different bacterial isolates that I had tested. There are 52 of them total. And on the y-axis is the percentage of larvae that have metamorphosed as a result of those bacterial isolates. Filter seawater, sterile filter seawater, is used as a negative control here on the right. And on the other end of the spectrum, on the left, is a natural biofilm. So this is a complex biofilm consisting of multiple species of bacteria that have been growing naturally in our sea tables over time. And natural biofilm itself is a pretty strong inducer, as it induces about 70% metamorphosis. And as you can see here, as far as the different isolates that were tested, there is a huge range. And so there are 3 in particular that are strong inducers relative to the positive control, and others that are moderately inductive, weakly inductive, and those that were non-inductive altogether. And when we look at these 52 isolates in the context of a phylogenetic tree here, with their closest blast matches based on their 16S gene sequence, we can look at the fact that there are several different types of genera represented here. And the 2 I'd like to focus on are Pseudoraltromonas and Thalassomonas, as the 3 strong isolates that I had in my previous graphs are highlighted in red here. And what I'd like to point out is that they belong to 2 different genera. And this indicates that not all species within a given genus are the same, not all of them are equally inductive. And this would indicate that whatever the actual substance is that makes bacteria inductive for coral settlement is not just isolated to one specific species, but it's common in at least 3 of them. However, there is a caveat in that in the previous experiment, I didn't take into consideration biofilm density. And shown here, I just want to show an example of this particular species, Pseudoraltromonas luteoviolacea. So it's the same species in both panels here. And these bacterial cells have been stained with propydium iodide and counted on a microscope. And on the left-hand side, this bacterium was inoculated at a density of 10 to the 7th cells per mil. On the right-hand side, there's a 10-fold dilution, 10 to the 6th cells per mil. And as you can see, this inoculation density is also reflected in the biofilm density, and that the biofilm on the left-hand side is much denser than the one on the right. And so the question becomes, how does this influence larval settlement then? And more specifically, if I go back to my 52 isolates and pick out 4 to look at, can I make these moderate inducers and this weak inducer a strong inducer if I were to increase the biofilm density? I mean, one would think so. Or if I were to dilute the strong inducer, can I make it a weak inducer? And so that's what I essentially tested. And so these are the different species, the 4 species that I looked at. Pseudoraltromonas is the strong inducer. Altromonas albanelli and Vibrio harvii are the moderate inducers. And Bacillus firmus is the weak inducer. And along the x-axis are the biofilm densities, and along the y-axis, the percentage of larvae that metamorphosed as a result. And the take-home message from this is that if you look at these 3 here, the Altromonas, Vibrio, and Bacillus, which are clumped together in this group in the middle here, even though they make much denser biofilms than the Pseudoraltromonas, this Pseudoraltromonas remains to be a strong inducer. And as far as the other ones, no matter how much you increase the biofilm density, it is still a weak or moderately inductive inducer. So this particular species, so going back to these questions here, is there specificity in the inductive capacities of different bacterial species? The answer is yes. And does biofilm density have an effect? And it's only for a strong inducer. So as far as ones that aren't strong to begin with, you can't make them strong by increasing their biofilm densities. What's really interesting about this particular genus here, Pseudoraltermonas, is that species belonging to this genus often induce an array of larvae, several different types of marine invertebrate larvae, in fact. And this includes the hydrozoan, Hydractinia, which is used in a lot of cnidarian development studies, 2 species of acropora coral, as well as barnacle, Balanus amphitrite. And this particular species, Pseudoraltermonas luteoviolacea, is also the same species that induces the larvae of an Australian sea urchin, as well as a biofouling tube worm, a polychaete known as Hydroides elegans. And so they don't just induce corals, they induce other invertebrate groups as well. And what makes this really interesting is if we can identify something like this, a particular bacterium that induces this wide range of marine invertebrates, we can potentially actually identify what the source is. What is it about the bacteria that gives it the cue? And so currently, my former lab where I did my graduate work is continuing on with this research as far as looking at this particular bacterium closer, looking at the genes that might be involved in its inductive capacity for inducing the larvae to settle and metamorphose. So next I'd like to move on to the other side of the story. How does the larva sense the cues? And first I'd like to show you a video clip here of what the larvae look like when they swim over these different surfaces. So these are coral planulae swimming over a clean surface. This is in a dish with just filtered seawater. There's no substratum at the bottom. And as you can see, they glide over pretty fast, not really taking the time to explore the bottom. Now if you look at this one here at the bottom, these are planulae over a biofilm surface. And I'll probably have to point them out because they're moving so slowly. But here's one here, here, one here which is practically sitting still, and one here as well. So they kind of blend in with the biofilm too, but you can see that they're definitely not moving around like crazy over a clean surface, for example. And so clearly there's a behavioral change here when you put them in a biofilm, that they are taking their time to really explore it and see if it's suitable substratum for them to make it a permanent home. And the question I'd like to focus on here is how do you go from the settlement cue that's in the substratum to the larva and the potential sensory cells that it utilizes to detect the cues. And this still remains a question, really. And in particular, I'm interested in which region of the larval body is directly involved in the detection of cues from the substratum. And if that region was removed, would larvae still be able to undergo settlement and metamorphosis if they can't detect it? And for this section, I focused on 2 coral species. One we're already familiar with, Postdoctoral Researcher, and the other one is Montipora capitata, and this is another common Indo-Pacific coral in Hawaii. And Postdoctoral Researcher, once again, is a brooder. Montipora capitata is a broadcast spawner. And so there are 2 different reproductive strategies that I'm also looking at here. And once again, Postdoctoral Researcher settles in response to bacterial cues in a natural biofilm. To talk a little bit about the life history of Montipora capitata. So this is a broadcast spawner that reproduces only during the summer months from June to August during the new moon. And they are hermaphrodites. And so they release these egg sperm bundles that contain about 14 eggs packaged together with the sperm in the middle. And upon reaching the surface of the water, they disintegrate pretty rapidly. And they have to mix with other bundles of a different adult colony because they can't self-fertilize. They would have to cross with other bundles of a different colony in order to produce viable larvae. And this is what their larvae look like. So they're a little smaller than the Postdoctoral larvae. They too also have the symbionts, that's that brown pigmentation in there, and these are vertically transmitted, just like in the other species. So they don't get them from the environment, they get them from the parent to begin with. And they also settle by flattening in the oral-aboral direction. And so this is a group of settled juveniles there with their mouths facing up. And in 2004, Steve Kolinsky found that this species settles in response to crustoscoralin algae. So there are several differences between the two species now in that they have different reproductive strategies. One larva is much larger than the other, and they have different settlement cues as well. And we'll see how all of those factors make a difference. So first, I employed immunohistochemistry to determine whether structurally I can see something different between the aboral end and the oral end, since what I said earlier, the aboral end is the end that attaches onto a substratum. Now, FMR-FMide is a common neuropeptide associated with sensory cells in a lot of other invertebrate larvae. So I used an anti-FMR-FMide antibody to stain the larvae of Postdoctoral to see whether I can differentiate any particular sensory cells at the aboral end. And this is a confocal image here on the left showing the epidermis. So this is lining the entire edge of the larva, and this is the interior of the larva, this is the exterior in contact with the environment. And as you can see, these FMR-FMide immunoreactive cells line across the entire edge of that epidermis. And this is just a different view. So this is looking at across the top, the surface of the larva. And here you see these cells as a network of cells, really. And cnidarians have simple nervous systems that contain a nerve net that transduces signals back and forth. And so it's not surprising to see this kind of network there. And the arrows are pointing to dendrites that are sitting on top of the cell bodies that are coming out towards the surface. And in terms of comparing whether there was a difference between the aboral end and the oral end, there really was no difference. These FMR-FMide cells were found across the entire larva, across the entire length of the larva. So they're probably not what I'm looking for, really. But it was a good place to start. And in going back to the literature,